Welcome back to a new video tutorial session from Biology Boost Up. Today in this video tutorial session we going to study about 10 tools used in genetic engineering or biotechnology. So now let's start. At first, the polymerase chain reaction or PCR. It is a rapid, versatile in vitro method for amplifying single molecule of DNA, involving repeated cycle of denaturation, hybridization to oligonucleotide primers, and synthesis of polynucleotide by the action of Taq polymerase at higher temperature. And here you can see the PCR machine, how it functions. Now look, PCR is known as polymerase chain reaction. It is efficient because it multiplying the DNA exponentially for each of the 25 to 75 cycles. A cycle takes only a minute, and each new segment of DNA that is made can serve as template for new ones. Next, the restriction enzyme. The discovery of enzymes known as restriction enzymes has been essential to protein engineering. Those enzymes cut DNA at specific location based on the nucleotide sequence. Hundreds of different restriction enzymes capable of cutting DNA at a distinct site have been isolated from many different strains of bacteria. DNA cut with a restriction enzyme produce many small fragments of varying size. Those can be separated using gel electrophoresis or chromatography. Now next, the gel electrophoresis. The gel electrophoresis is a process by which we can separate the fragments of DNA which is made by after the reaction with restriction endonuclease according to the size of the fragments. Now look, purifying DNA from cell culture or cutting it using restriction enzymes would not be of much use if we could not visualize the DNA. That is, find a way to view whether or not your exact contains anything or what size fragments you have cut it into. One way to do this by gel electrophoresis. Gels are used for a variety of purposes, from viewing cut DNA to detect cutting DNA inserts and knockout. Now next, the DNA ligase. The DNA ligase catalyzes the covalent closure of nick in DNA molecule by using energy from NAD or ATP. In genetic research, it is often necessary to link two or more individual strands of DNA to create a recombinant strand or close a circular strand that has been cut with restriction enzyme. Enzymes called DNA ligase can create covalent bond between nucleotide chains The enzymes DNA polymerase 1 and polynucleotide kinase are also important in this process for filling in gap or phosphorylating the 5' ends respectively. Next, the polymerase. The polymerase enzyme helps to polymerizations of nucleotides as well as it acts as a proofreading action. The groups of enzymes that catalyze the synthesis of nucleic acid molecule are collectively referred to as polymerase. It is customary to use the name of the nucleic acid template on which the polymerase acts. The three important polymerases are given below. First, DNA-dependent DNA polymerase that replicates the DNA from a DNA. Second, RNA-dependent DNA polymerase or reverse transcriptase that transcribe DNA from a RNA, and DNA-dependent RNA polymerase that transcribe the RNA from a DNA. Next, the prokaryotic host. The host are those organisms inside which the recombinant DNA replicate itself, and this is the electron microscopic view of E. coli bacteria. The bacterium E. coli was the first organism used in the DNA technology experiments and continues to be the host of choice by many workers. Undoubtedly, E. coli, the simplest gram-negative bacterium, has played a key role in the development of present-day biotechnology. Under suitable environment, the number of E. coli can double every 20 minutes. As the bacteria multiply, their plasmid along with foreign DNA also multiply to produce millions of copies referred to as a colony or in a short colony. The term colony broadly refers to a mass of cell, organisms or genes that results from the multiplication of a single cells or genes. Next, the eukaryotic host. That means where the eukaryotic cells act as a host and inside which the recombinant DNA replicate itself. Now look, eukaryotic organisms are preferred to produce human proteins since those hosts with complex structure are more suitable to synthesize complex protein. The most commonly used eukaryotic organism is the yeast. It is a non-pathogenic organism routinely used in brewing and baking industries. Certain fungi have been also used in gene cloning experiments. Now next, the replicating DNA. The replicating DNA, also known as the carrier molecule or vectors, having the capability of independent replicating itself. Small circular pieces of DNA that are not part of bacterial genome but capable of self-replication are known as the plasmid. Plasmids are often used as vector to transport gene between microorganisms. In biotechnology, once the gene of insert has been amplified and both the gene and plasmid are cut by restriction enzymes, they are ligated together, generating what is known as recombinant DNA. Viral DNA can also be used as a vector, as can plasmids, recombinant plasmids containing bacteriophage genes. Next, the transformation. Transformation of the recombinant DNA molecule carrying an inserted DNA fragments into a host cell in which the chosen molecule can undergo replication independently of the host cell chromosome and produce dozens of identical cells known as clone. Now look, the process of transferring genetic material on a vector such as plasmid into new host cell is called transformation. This technique requires that the host cells are exposed to an environmental change which makes them competent or temporarily permeable to the vector. Electrophoresis is one such technique. The larger the plasmid, the lower the efficiency with which it is taken up by cells. Larger DNA segments are more easily cloned using bacteriophage, retrovirus or other viral vectors or plasmid in a method called transduction. Phage or viral vector are often used in regenerative medicines but may causes the insertion of DNA in parts of our chromosome where we do not want it, causing complication and even cancer. Next, the methods of select transgenic organism. Not all cells will take up DNA during transformation. Therefore, it is essential to identify the cells that undergo a transformation and those that have not. Generally, plasmid carry genes for antibiotic resistance and transgenic cell can be selected based on the expression of those genes and their ability to grow on media containing that antibiotic. Alternative methods of selection depend on the presence of other receptor protein such as the X-galactose or LAC-Z system or green fluorescence protein, which allow selection based on color and fluorescency respectively.